Welcome back to the course Fundamentals of General Immunology. So we understood the lineages, how these lineages are committed to do different functions. So today we will try to understand about the functional plasticity of the immune cells. In general, plasticity stands for how the things are changed in due course of time when there is a change in the environmental structure of an immune cell. So we'll try to understand that in details in today's lecture about how this functional aspect of plasticity modulates on immune cells. So by definition, the immune cell plasticity refers to the dynamic ability. So it's a dynamic process. It can't be a staggered or it can't be a static process. It's a dynamic process where the immune cells are altering their phenotype. Now when we say phenotype, there are two terms which we generally discuss much oftenly in biology. One is called genotype, which is makeup of a cell, where a cell is made up of a particular kind of a genetic material. That's what you call genotype. And the outward manifestation of this genotype is what is visible to our eyes. That's called phenotype. So the phenotype is basically the things which are visible to our naked eye, and the genotype is one which is already embedded into the cell in the form of a genetic information. So generally phenotype is changed based on the environment, based on the metabolic state of the cell, and that is what we refer to the plasticity of the immune cell. So generally their phenotypes are altered, their function and metabolic states are altered in response to the environmental stimuli without changing the genotype. That's what the complete definition of immune cell plasticity. So let's repeat it one more time. So it's a dynamic ability of the cells, particularly the immune cell, and we will try to note down the critical points which are there, such as dynamic ability of an immune cell that can lead to alteration in their phenotype, their functions and the metabolic states based on the environmental stimuli without changing much on the genotype. And that is what we understand about the immune cell plasticity. Now this flexibility is driven by, so basically which we understood that there are environmental factors which are responsible for it. So this flexibility is driven by metabolic and epigenetic shifts. Now let's try to understand the metabolism. Metabolism is what a cell do. This is a combination process of anabolism and catabolism, where a constructive and the destructive process together forms a metabolism. So if there is anything change upon the environmental stimuli to alter the metabolism or the epigenetic shift. Now epigenetic shifts are actually, definition is given in the bottom. So epigenetic shifts are actually, the definition is given in the bottom. So epigenetic is a study of how environment, the lifestyle and the behavior influence the gene expression, whether the gene expression is turned on or turned off without changing the underlying DNA sequences, which is the genotype. So basically epigenetics are the specific study where the gene expression are turned on or off based on the environmental stimuli, and the two classical examples are DNA methylation and the histone modification. We know that in the nucleus, inside the nucleus we have a DNA material which are bound tightly with this protein called histone. These histones are positively charged proteins, and since these are positively charged and the DNA is negatively charged, they are in the tight bound condition inside the nucleus, and any alteration in the histone in terms of its modification could lead to alteration in the DNA binding capacity of the histone, which could lead to the gene expression turn on or turn off based on the sequence specificity. So this is very important process, and again the DNA methylation. So the way the histone is modified, similarly the DNA can also be modified, where the charge of the DNA can be altered because of the addition of methylation, and the DNA methylation could vary the gene expression in a different style. So essentially this immune plasticity which we are discussing about the change in the phenotype without changing in the genotype could be because of the environmental factor, which could lead to alteration in the metabolism and also the genetic shift, allowing cell to alter the gene expression and remember the previous stimuli. So essentially, we will try to understand how this plasticity of the immune cell could vary in their response without changing the genotype, and they can do individual functionality for some distinct pathways and do an entirely different function altogether. And this plasticity of an immune cell is very important when it comes to alteration in the cell type, such as could be a different kind of macrophages, there could be a different type of neutrophils. They are same genotypically, but their functions are different. So we'll try to understand those in details in coming slides. So let's move on. And we understood that the plasticity, the cellular plasticity, is actually a process where the cells are phenotypically changed. And let's try to understand the phenomena in context of little bit of the energy kinetics. Now say for example, these are the stem cells which are getting differentiated or de-differentiated into some progenitor cell mass, and these progenitor cells are ultimately finally differentiated into the cell type which can do functional. So these are actually a stem cell to progenitor cell and to differentiated cell mass which can do some functionality. Now the plasticity, the cells could be a very high in terms of plasticity and the cells are very low in terms of plasticity. When we say that the cells are very high in plasticity, that means the cell can alter into a different phenotype very easily. If the things are changed very easily, that means the energy requirement to change that cell type will be very less or minimal. And in that case, suppose if there is a cell A which can change to the cell type B, so that there is a change in the plasticity of a cell. Now since this is a very high plastic cell, we see that the energy barrier is very less, and therefore the cells can easily do a transformation from one cell type to other cell type very easily without changing the genetic information. While in case of low plastic cell, that means the cells are very rigid, they can't change very oftenly. That means a cell type A may change to cell type B very rarely. It's not very often a process, and therefore the energy barriers are very high. So you require to have a very high amount of energy to be supplied to the cell type A in order to convert into B cell type. Well, that's not the case when the cells are highly plastic in nature. So it all depends upon how energy barrier can be moved from one location to other location to change the cell type from one to other cell type. And we could understand it in terms of analogy that if there are high plasticity, the barriers are less, and if there is a low plasticity, that means the barrier is really high. You need to provide really high energy, or kind of a processes required is quite cumbersome in order to change the cell into a different cell type. Now there are two terms which are here listed called transdetermination and the transdifferentiation. Now remember, these two terms are related to different cell population. So transdetermination is the process by which distinct cell type but related population of a stem or progenitor. So one can easily see that the transdetermination terms are very specifically to stem cell or the progenitor cell. That means this term is applicable to these two only, the stem cell and progenitor cell, which can convert into one type or other type. For example, when we take a Drosophila as an example, so Drosophila is a fruit fly where the cells can be defined in very embryonic stage to form a particular cell type, but when we transplant those cell type into a different Drosophila embryo, we see that there could be a modulation in the cell type where the leg cells can form the wing and vice versa if there is a transplantation of the cell type. So this is what we call transdetermination, which is very specifically to cell type, which are either stem cell or the progenitor cell. While the second term is transdifferentiation. Now transdifferentiation is a term which is typically used to the differentiated cell pass. So once the cells are differentiated, that means cells can't go back to the its progenitor cell type. So therefore, we always see that there is a transdifferentiation that's happening mostly in the cancer cell population, where there is a term called epithelial mesenchymal transition in multiple cancer type, where a particular cell type like epithelial cell can be converted into mesenchymal cell and vice versa. The other processes could also lead to different forms of cancer or metastatic alteration in the cancer cell. So this is a typical process which is related to defined differentiated cell mass. So this is actually a nutshell about the cellular plasticity, that keep in mind the two terms, that if the cells are high plastic, we need low energy barrier, and if it is low plasticity, then we need a high energy barrier. And particularly these two terms, transdetermination and transdifferentiation, which are very specifically in category for the different cell types. All right, we'll move on to a separate topic when we discuss about the cellular plasticity and how the cellular plasticity is altered by epigenetic and environmental stimuli. Let's try to understand the cell specific type wise what are the plasticity. So let's understand the plasticity on macrophages. So macrophages, we know these are specifically antigen presenting cell which can present the antigen to the immune cell so that the immunity can be raised in a proper way. And if we start looking into the macrophages, there are two types of macrophages. One is called M1 macrophages and the other is called M2 macrophages. We will see how these M2 and M1 is differentiated from the M0 macrophages in the next slide. But for understanding here the plasticity of the two distinct cell mass, the cells are same, the phenotype is different, and therefore we can categorically call it one as an M1 and the other is an M2. So the M1 macrophages, actually if you look into the function first, so this is a pro-inflammatory activity. This can lead to microbial and tumoral activity. It can lead to tissue damage. So therefore these are related to some kind of harmful processes. So involvement of M1 macrophages is towards the pro-inflammatory side. We can see if they are involved, we can understand that there is a harmful processes going on. While in case of other side, when we have an M2 macrophages, this is anti-inflammatory. So it can stop the inflammation. We'll see how it can stop the inflammation. It can also do a phagocytotic activity. That means it can engulf the particulate or other form of antigen when they are entering inside the body. They can involve in tissue regeneration and repair. That means they are very good when there is a damage post inflammation. We can see that either the tissues are repaired or there is a regenerative capacity that can be regained because of these M2 macrophages. They are involved in angiogenesis. Angio stands for blood vessel, genesis means formation. They can help in blood vessels formation. They can also involve in immunomodulation, tumor formation and progression. So basically these are basically an involvement where we can see that these are typically done in a different way, but majorly we can focus here that these are anti-inflammatory in function. Now when we see the M2 macrophages and how these are different than M1 macrophages, we could understand that there are surface expression of the proteins, and these proteins are very different in terms of their phenotype. So one can see that there are specific CD molecules such as CD163, CD206, CD209, FIDG1 receptor, YM12 receptor. And on the other hand, when we see M1 macrophages, these are CD80, 86, toll-like receptor 2, toll-like receptor 4, MSC2 and INOS, which are basically a very unique set of molecules involved in inflammatory processes. Now, if we see that these are two distinct phases, there could be an alteration in the gene level also. So we understood that typical phenotype of cell is decided by what kind of transcription factor, what are the genes expression that's happening in the cell type. So we could see that the gene expression is entirely different here, such as STAT3 and 6, while here in case of the M1 macrophages, we could see that we have NF-kappa beta, STAT1, IRF3, and some other specific transcription factors. That means this modulation of the gene expression is turned on upon the expression of the very specific transcription factor, which can lead to entirely a different macrophages phenotype. So we'll try to see it in a different context when we discuss how these macrophages are generated. So we could see that the macrophages, these are M0 macrophages, and upon the and these are non-polarized. Non-polarized stands for their functionalities are not defined. So if we can have an M1 polarization, we can see that these are the M1 macrophages, and when there is a diversion towards the M2 macrophages, then see the polarization is different. M2 further is divided into M2A, M2B, M2C, and M2D depend upon their function. For example, the M2A is responsible for tissue repair, wound healing, anti-parasitic response, and we could see that the production of cytokines are also very different from these cell types. Secondly, the M2B is again having a separate set of receptor protein which are expressed on their cell surface. The function are different, for example, immunoregulation, Th2 activation, a mixed cytokine profiles, and also the production of entirely different set of cytokine altogether. So the cell specific phenotype will largely do a functionality based on what kind of cytokines they are producing, what kind of cell proteins they are secreting out into the environment, so that they can see a different kind of gradient altogether. For example, M2C, we can see that IL-10 and TGF-beta is getting out from this cell type, which can lead to tumor remodeling, apoptotic cell clearance, and immunosuppressant, stuff like that. And again, M2D is involved in tumor progression, immunosuppression, and again these are controlled by secretion of different set of cytokines. So one can understand that the polarized cell population, which are largely a plasticity of a cell, immune cell, are very, very versatile, very divergent cell type are produced upon the stimuli, upon the gene expression, upon the environment and all. And this could critically delineate the pathway of each cell type that can do a function in a different set of immune regulation. So we could understand that this is just an antigen presenting cell plasticity, where two cell populations such as M1 and M2 can do a different set of function altogether based on their cytokine profiles and their expression of the protein on their cell surface. So let's try to understand a separate cell type, and that's neutrophil. Now neutrophils are the cells, again very, very important for the phagocytosis process, because this cell can kill the pathogen by cytotoxicity or the production of specific reactive oxygen species, and do some function related to the immune, and do the function based on their specialty. So when we try to see that what are the different type of cells which are present when we talk about neutrophils. So neutrophils are again divided into two types called N1 and the N2. So N1 is actually anti-tumoral, which is good, and N2 is actually pro-tumoral. That means they can support the tumor progression, and one can see that there could be a TGF-beta specific regulation, and here there is an interferon beta specific regulation. So when there is an interferon involvement, we could see that since it's a non-specificity, it's a non-specific immune response, we can see that there are involvements such as cytotoxicity, the tumor rejection, and also the immune memory. So one way or other way, we can see that the interferon specific involvement of N1 can lead to the tumor suppression or anti-tumoral activity, while in other case we have TGF-beta, which is called transforming growth factor beta, that could lead to the invasion, which is what is critical in cancer research, such as metastasis, and also causing downregulation of the immune response, so that the body may not understand that these cells are not in true sense the self. So there could be an immunosuppression. the tumor growth will be increased, and of course when there is more and more cell population, we need to provide more and more nutrition to the cells or the blood supply. So we could see that directly there is a environment of blood vessel formation, which is called angiogenesis. So this N2 is actually helping all these processes in order to say progress the cancer. So N2 involvement in the body immune system is harmful, while in case of N1 phenotype, we could see that the N1 is antitumoral. So when we decide that what kind of immunity we need to take care, we need to think about plasticity of the cell very carefully. So we want to have N1 in a higher concentration as compared to N2 in order to give a positive effect towards the tumor environment. So, and again, of course, TGF-beta could be an inhibitor. This sign indicates inhibitor in biology, and also here the interferon beta is an inhibitor. So both way we can see that one is progressing the stuff and the other is inhibiting the stuff. So these are again very critical plasticity of the neutrophils which exist apart from macrophages. So let's try to move on and see the plasticity of the T helper cell population. So we understood when we discussed about the major histocompatibility complex, and when we discussed about the major histocompatibility complexes, we understood that if there is an MHC2 specific response, then we can see that this can lead to CD4 specific immunity, or this is also called a T helper cell population stimulation. So T helper cell is again showing a huge number of plasticity, and let's try to understand this critically when we see how these cell population are varying in their function. So say once the antigen presenting cell is processing the antigen through MHC2, then the CD4 specific T cell is coming into picture and getting activated, and upon activation we could see that there are a lot of cytokines which are secreted, and these cytokines can decide the fate of a cell type. For example, if there are interferon gamma and IL-12 production, we can see that the Th1 subsets are produced, which can lead to again interferon gamma and TNF alpha production. So that can produce more amount of interferon gamma and TNF alpha. While if there is a more production of say IL-2 and IL-4, we can see that the cells are polarized towards the Th2 population, and this Th2 can upregulate IL-4, IL-5, IL-9 and IL-13. Now when we say that these are multiple cytokines which are secreted upon the activation of a cell type, we need to understand that these are very critically controlled in the level of gene expression, and there are multiple pathways which control the expression of these cytokines. So similarly, if the IL-6, IL-23 and TGF-beta in combination or alone can lead to a different subset altogether, such as Th17. Again, Th17 is a very important T cell population that can lead to production of Th17 and IL-22. And then finally, if we can get more expression of IL-10 and TGF-beta, we could lead to production of T regulatory cell population, which can produce IL-10 and TGF-beta. Again, these are very critical cell involved in tolerance, particularly the oral tolerance. So we'll discuss about this T regulatory cell population when we discuss more about the immune tolerance and related aspect, how the immune response are not produced against the self antigen. So the T regulatory cell population does play a very important role in that aspect. So just to summarize the TH plasticity or the T helper cell plasticity, that once the cells are stimulated by an antigen presenting cells by a various pathway, we'll try to see this CD80 and 86 stimulation to CD28 molecules when we discuss in our next module about co-stimulation. So I'm not going to discuss this one here, but we'll see it in our future classes. But for time being, just emphasizing the point that the antigen presenting cell which are expressing the antigen through MHC2 can upregulate the CD4 specific T helper cell population, which again produce different kind of immune regulatory cytokine that can decide the fate of a cell type. And based on the different cytokine produced, the cells are polarized into Th subsets, which are called Th1, Th2, Th17 and T regulatory cells. And these cell type again in turn secretes multiple different cytokines such as interferon gamma, tumor necrosis factor alpha and other interleukins as listed here in this figure, such as IL-4, 5, 9, 13, 17, 22 and IL-10 TGF beta here in T regulatory cell as well. So this typical pattern of T helper cell plasticity decides again the fate of the immune cells in a great extent. So till now what we understood that there could be a plasticity in the macrophages, there could be a plasticity in the neutrophils and also in T helper cell. So there could be another subset of the cell which can also show plasticity apart from the tumor cell. So let's take another example and let's see what you call the innate lymphoid cell plasticity. And if you can recall back, we have discussed that there are individual cell types which are present in the human body, and this can inbuilt, they are not activated they are not activated upon the exposure to antigen. These are present inbuilt. So these are innate, these are present right from the birth, and these are lymphoid cells. So we can simply call it ILC, the innate lymphoid cell. So we will try to understand the plasticity of these innate lymphoid cell, and we can see that there are ILC1, C2, C3 and Creg. So these are in the same nomenclature, is exactly same like Th cell types, but we could see that their stimulating cytokines and other things are quite different. So the cytokines which are responsible for stimulating the production of these cell types are different, such as interleukins listed here, and also some of the cytokines here. Here also the one which can lead to the ILC3 production and TGF beta. Of course we don't know exactly which cytokine does this, but ultimately the interferon gamma is actually produced by ILC1, while IL-4, 5, 9 and 13 is secreted by ILC2, 17 and 22 is ILC3. So we can see that these are exactly the same line as what we discussed in the T helper cell. Again, these cells are present right from the innate. So the T helper cell population is coming into picture when there is an exposure to the antigen. So these are the first cell which come into the picture when there is an antigen encounter, and later on when there is an adaptive immune system, then we see a different Th subset population coming into picture. So these are, we can say that these are the first line, and the Th subset population plasticity coming in the second line of say involvement. So this is the function listed here. Again, there could be an immunity to the intracellular pathogens such as the parasite which are present inside the cell, such as viruses, and again there are disease associations such as Crohn's disease and COPD, which is again a pulmonary disorder. Then again in case of C2 cell, we can see that these are specifically the immunity related to helminthic parasite, wound repairs and all. So atopic dermatitis, asthma or chronic rhinosinusitis are one where we can see the involvement of this ILC2 cell population, while psoriasis, IBD and multiple sclerosis are exactly the target for ILC3 cell population, while the function of ILC regulatory cell population are not clear, but people have shown that they have a very high role in the colitis, which is an inflammation of colon, and we can see that large structure of T regulatory cell population are mostly towards the gastrointestinal tract. In T helper cell population also, when we discuss about the T regulatory cell population, our discussion will direct towards mostly towards the T regulatory cell population. So we could see that the T regulatory cell population are mostly towards the gastrointestinal cavity. So this is all about the innate lymphoid cell plasticity. So there are 4 cell types exactly like T helper cell types, and this can do an innate cell surveillance with the same function as what reported for the T helper cell later on when they are encountering the antigen by MHC2 population. So let's try to understand a very different way of plasticity, particularly in the tumor cell. Now the tumor cell environments are very different, and we need to understand that the plasticity of immune cell can modulate the tumor cell microenvironment as well. So let's try to see in this figure that suppose there is this cell mass where you have the complete cancerous cell which are there, and then just the cell mass is floating in an area, and these are just before the therapy. When there is a therapy induced, then the different kind of cytokines such as TGF-beta, the hepatocyte growth factor, IL-6, TNF, tumor necrosis factor, and other kind of cytokines come into picture, and accordingly we can see that the tumor cells are either dead or there could be a change in the plasticity of the tumor cell differently. So we can see that there is a switch in the phenotype. So these are cancerous cell mass, and this change in the phenotype can happen because of these cytokines which are secreted upon the treatment. So the immune cells are recruited, and this can lead to polarization. So what is happening that when we have a tumor cell, the tumor environment is very, very different. And there could be a specific the endothelial cell area where the blood cells are present, and once there is a treatment, the cell type can be changed, their phenotype based on the cytokine secreted by the immune cell. So once the treatment is there, the immune cells are coming into, say these are the immune cells which are coming into the site of the tumor in order to treat the tumor, but then these can start secreting this cytokine. Because of these cytokines, the tumor environment is changing themselves into a plastic form. So they are changing their phenotype in order to survive into this hostile environment. So this environment is not good for the tumor cell, and therefore the tumor mass is changing themselves into a different plastic form so that they can survive. And therefore once there is a more and more production of the cytokine, there is a chance of the relapse, because the cell mass is now changed phenotypically in order to escape from the treatment. And this is what is very common when we are talking about the cancer treatment. So once there is a treatment of cancer, we can see that the immune cells are recruited, started secreting cytokines, and these cytokines lead to polarization and plasticity of the tumor mass into a cell type which can escape the treatment, and ultimately leading to relapse by metastasis. So this is a very important say biology of the cancer cell when it comes to the plasticity. So of course we are trying to talk about the immune cell plasticity, but we need to understand that this plasticity is a general phenomena. When we are talking about a term plasticity, it is not only immune cell, this is related to all other cell type as well, and somewhere it is helping the body system to fight against the pathogen, and some places it can help the particular cell type to escape from that environment to find out something suitable for them. Just for here in case of tumor cell, because of the hostile environment of the cytokine and the immune cell, there could be a chances of relapse. So this is just to give a brief idea about how these immune cells can modulate the plasticity of the tumor cell. So let's try to understand finally that how this metabolic reprogramming can derive the functional shift in the cell type. So we understood that the cells are shifting from one phenotype to other phenotype, but what phenomena, what kind of molecular mechanism and detail can lead to deviation of this phenotype, that we need to understand. So suppose if we take just example here that when there is a T cell activation, the T cell activation can lead to a production of mTORC-C1. So mTORC-C1 is actually a mechanistic target of rapamycin complex 1, which is a serine threonine protein kinases. When we discuss about kinases, kinases stands for any protein that can do phosphorylation, so add phosphate group to a separate molecule, say X. So if this is a kinase, the function of this kinase is to add the phosphate group into another protein by utilizing ATP and converting this ATP into adenosine diphosphate, providing a phosphate group to this molecule. So this is very, very important kinase which is based on the serine threonine protein kinase complex that acts as a master regulator for the cell growth, proliferation and metabolism by sensing nutrient, energy and redox signaling. So when there is a nutrition deprivation or when there is an alteration in the nutrition switch or say if there is a lack of energy such as adenosine triphosphate or any kind of glucose metabolism and all, we see that this mTORC1 pathway comes into picture, where the mTORC1 can phosphorylate another downstream protein. And in this example, what it does is it can do phosphorylation of DDX21. DDX21 is just a dead box helicase protein. When we say helicase, the function of helicase is to remove the helical form of the structure into a single stranded molecules. For example, if it is a RNA helix, it can remove the helix so as the RNA can be accessible to other molecule for the functionality. So DDX21 is one such dead box protein, which is again a conserved enzyme that can use ATP hydrolysis to remodel the RNA protein complex and also unwind RNA duplex structures in order to start the functionality. So what we are trying to understand that how this metabolism is modulating the shift in the cellular plasticity is, suppose there is an activation of the T cell population. If there is a nutrition deprivation or energy starvation or any kind of redox related stress, the mTORC1 comes into picture, which is a kinase, so it does a phosphorylation. And once this is a phosphorylation, then we see that the phosphorylated DDX1 can sit over this specific element called ARE. ARE stands for AU rich element, and these are the regulatory sequences around the 3' untranslated region, right? So these are present in the 3' end. So we have always the messenger RNA where the 5' is 7-methyl guanosine cap, and then we could have some untranslated region. Again, there could be a coding sequence right from the ATG to stop codon, and finally we have a regulatory sequences. So ARE is actually AU rich element which are present in the 3' site of the untranslated region, and they can have a very dynamic role in controlling the gene expression or the RNA stability along with the translation rate, because again the mRNA is what will form the protein downstream. So what's happening is actually we could see that the post activation of the DDX21 phosphorylation, this can dock into the ARE site, and this can lead to the production of some of the interleukins, for example IL-2, which can further cause the cell type to release different cytokines which can modulate the cellular function. So ideally we can see that these all functional shift in the plasticity is controlled very tightly by the gene expression. And we just gave an example of mechanistic target of rapamycin complex 1, which is actually a kinase that can phosphorylate another protein to control the protein expression and cytokine regulation on a specific T cell population. And by doing this, it can control the cellular homeostasis in the immune cells. And these are just one example. There could be multiple different examples that can lead to the deviation of the plasticity or the cellular functionality upon the metabolic reprogramming. So this is all about the cellular plasticity. We'll try to understand the different aspect altogether, that how this receptor editing and other things are happening in our next class. Thank you.